Clinical presentation of mast cell activation syndrome. Until 2007, our understanding of the molecular genetic roots of systemic mastocytosis was relatively simple. Most cases, more than 90%, appeared to be driven by any one of a small set of mutations clustered, for the most part, around KIT codon 816, site of one of the molecule's two kinase, i.e. activating, domains, framing the actual enzymatic site. Most mutations in this region seem to cause constitutive activation of the KIT protein. Thus, binding of ligand stem cell factor is no longer needed to activate the multiple pathways downstream from KIT, which can result in reproduction, anti-apoptosis, which has turned out to be the dominant manner of mast cell accumulation in mastocytosis, differentiation, and mediator production and release. Poorly explained by this relatively small array of mutations, though, was the marked clinical heterogeneity of mastocytosis. To be sure, certain remarkable features which had long been the clinical hallmarks of mastocytosis, e.g., idiopathic anaphylaxis, odd and copious allergies, spontaneous flushing, vasomotor instability, diffusely migratory rashes and edema, were present in most cases. But many other symptoms and findings were noted, too, with little consistency across this patient population. In 2007, the seminal findings of Molderings et al. Revealed the likely answer for this heterogeneity. A much wider array of presumably activating mutations, scattered across all domains of KIT, was detectable by whole KIT mRNA sequencing in most mast cell activation syndrome patients. In fact, many patients appeared to bear in KIT not just one mutation, but in fact multiple mutations. These findings were repeated and extended by Molderings et al. In 2010, and other groups simultaneously were identifying activating mutations in other mast cell regulatory elements too. By 2011, it was readily apparent that the chronic myeloproliferative neoplasms, including mast cell diseases, often bear multiple mutations in the regulatory elements in the neoplastic stem cells, with evolving genomic disruption often heralding clinical progression. Over the last 70 years, our understanding of mast cells' mediator content has dramatically evolved too. From a time when the known and possible mast cell mediators numbered a mere handful, the body of research to date has identified that in truth, mast cells produce and release scores of mediators. Modern estimates range from more than 60 to more than 200, many of remarkable potency. Given the mast cell's fundamental role as a vehicle for production and release of these mediators, it is not surprising that mutations in the cell's regulatory elements lead to aberrancies in such production and release. Although most of these mutations investigated to date appear to cause constitutive mast cell activation, leading to increased mediator production and release, loss of function mutations could be pathologic as well. As all mast cell mediators are intimately evolved in various aspects of homeostasis of all of the body's cells, tissues, organs, and systems, it should not be surprising that either too great or too little production and release of any given mediator likely will have consequences, first at the cellular level, and ultimately at the clinical level. Thus, with any one mutation likely resulting in aberrant production and release of many mast cell mediators, and with most mast cell diseases harboring multiple mutations across all domains of many or all of the cell's regulatory elements, it should be expected that mast cell activation disorders will present with an extraordinary degree of clinical heterogeneity to match the extraordinary degree of underlying mutational heterogeneity and resulting heterogeneity of patterns of aberrant mediator expression. Presented below are the clinical findings, system by system, which have been observed in mast cell activation syndrome patients, both in the peer-reviewed literature, cited as available, and in the author's experience with more than 300 such patients diagnosed since 2008.